Right, now, good morning to all of you. Welcome back to for a continuation of yesterday's lecture. It's early morning here in India, and the time is 4:50. We are dealing with the second chapter on cell structure and organization. And we were on the topic of transposons. So with a little portion left out there, we're just going to cover up that and then move on to the next section. Right, so we just about finished. We were mentioning about the signs and lines. So, and similar to other mobile elements, now most signs have accumulated mutations from time of the insertion in the germline of an ancient ancestor of modern humans. Like lines, many signs are also truncated at their 5' end. The table summarizes the major types of interspersed repeats derived from mobile elements in the human genome. The table shows class, length, copy number in human genome, and the function of human genome. For DNA transposons, length is 2 to 3 kb, and the copy is 300,000, and the fraction of human genome percentage is 3. LTR retrotransposons, the length is 6 to 11 kb, the copy is 440,000, and the fraction percentage is 8. Non-LTR retrotransposons, we have lines that are 6 to 8 kb, 860,000 in length, that's the copy number, and a fraction percentage of 21. Signs with a length of 100 to 300 pp, and a copy of 1.6 million, and the fraction percentage at 13. Well, in addition to the mobile elements listed in table DNA, copies of a wide variety of mRNAs appear to have integrated into chromosomal DNA. Since these sequences lack introns and do not have flanking sequences similar to those of the functional gene copies, they clearly are not simply duplicated genes that have drifted into non-functionality and become pseudogenes as discussed earlier. Instead, these DNA segments appear to be retrotransposed. Copies are spliced in polyadenylated processed mRNA. Compared with normal genes encoding mRNAs, these inserted segments generally contain multiple mutations, which are thought to have accumulated since their mRNAs. We first reverse transcribed and randomly integrated into the genome of a germ cell in an ancient ancestor. These non-functional genomic copies of mRNA are referred to as processed pseudogenes. Most processed pseudogenes are flanked by short direct repeats, supporting the hypothesis that they were generated by rare retrotransposition events involving cellular mRNAs. Now, once we finish this, at the end of each chapter, right, we'll go for the qRNAs, but then as each topic I finish the lecture on the succeeding lecture, I'll try my best to give you Q&As. We'll put about 4 or 5 Q&As of the previous, then go to the next chapter, okay? And whatever is being covered additional, Q&As later on the next topic, so that you get to know what the subject is about and you don't get that kind of confused, but at the same time you get enough time to listen to the lecture as it's being delivered to you, plus you can listen to the Q&A, right? Okay. Carrying on here, other moderately repetitive sequences representing partial or mutant copies of genes encoding small nuclear RNAs, snRNAs and tRNAs are found in mammalian genomes. Like processed pseudogenes derived from mRNAs, these non-functional copies of small RNA genes are flanked by short direct repeats and most likely result from rare retrotransposition events that have accumulated through the course of events. Enzymes expressed from a line are thought to have carried out all these retrotransposition events involving mRNAs, snRNAs, and tRNAs. Right? Mobile DNA elements probably had a significant influence on evolution. Although mobile DNA elements appear to have no direct function other than to maintain their own existence, their presence probably had a profound impact on the evolution of modern-day organisms. As mentioned earlier, about half the spontaneous mutations in Drosophila from insertion of a mobile DNA element into or near a transcript unit. In mammals, however, mobile elements cause a much smaller proportion of spontaneous mutations. And about 10% in mice and only 0.1 to 0.2% in humans. Still, mobile elements have been found in mutant alleles associated with several human genetic diseases. In lineages leading to higher eukaryotes, homologous recombination between mobile DNA elements dispersed throughout ancestral genomes may have generated gene duplications and other DNA rearrangements during evolution. For instance, cloning and sequencing of the beta-globin gene cluster from various primate species has provided strong evidence that the human G1, G gamma and A gamma genes arose from an unequal homologous crossover between two L1 sequences, flanking an ancestral globin gene. Subsequent divergence of such duplicated genes could lead to acquisition of distinct beneficial functions associated with each member of a gene family. Unequal crossing over between mobile elements located within introns of a particular gene could lead to the duplication of exons within that gene. This process most likely influenced the evolution of genes that contain multiple copies of similar exons encoding similar protein domains, such as the fibronectin gene. Some evidence suggests that during the evolution of higher eukaryotes, recombination between interspersed repeats and introns of two separate genes also occurred, generating new genes made from novel combinations of pre-existing exons. This evolutionary process most likely influenced the evolution of genes that contain multiple copies of similar exons encoding similar proteins domain, such as the fibronectin gene. Some evidence suggests that during the evolution of higher eukaryotes, recombination between interspersed repeats in introns of two separate genes also occurred, generating new genes made from novel combinations of pre-existing exons. This evolutionary process, termed exon shuffling, may have occurred during evolution of the genes encoding tissue plasminogen activator, the new receptor, and epidermal growth factor, which all contain an EGF domain. In this case, exon shuffling presumably resulted in insertion of an EGF domain encoding exon into an intron of the ancestral form of each of these genes. Both DNA transposons and line retrotransposons have been shown to occasionally carry unrelated flanking sequences, which, when they insert into new sites by the mechanism diagrammed in Figure 11. These considerations suggest that early view of mobile DNA elements as completely selfish molecular parasites misses the mark. Rather, they have likely contributed profoundly to the evolution of higher organisms by promoting 1. The generation of gene families via gene duplication, 2. The creation of new genes via shuffling of pre-existing exons, and 3. Formation of more complex regulatory regions that provide multifaceted control of gene expression. Right? So each topic will be noted, all right? And at the end of the session of each, like I've been telling you about the other chapters, we'll have the Q&A, right? Exclusive Q&A. Let the portions be completed first, right? So both would be hand in hand, very helpful. And at the end of each day, as we finish, we'll once we sum up, on the next day before starting, we'll have a Q&A. Like that, likewise, from now on, each day, we have Q&As, right? On the previous chapters that is just completed, right? Okay, and in between, when you have doubts, you could always send me a query or any kind of questions that you have. Based on the subject, you could send your queries and for any clarifications to my email or to my YouTube comment section, right? Come to that section and on the comments, we can interact. Now the next section will be D, that's cell division and cell cycle. Systems of cell division. 4 events occur before and during cell division. A signal to reproduce must be received. Replication of DNA and vital cell components must occur. DNA must distribute to the new cells. The cell membrane and cell wall in some organisms must separate the two new cells. Prokaryotes divide by fission. Prokaryotic cells grow in size, replicate DNA, and divide into two new cells. This process is called fission. Escherichia coli, a bacterium, simply divides as quickly as resources permit at 37 degrees centigrade. This is about once every 40 minutes. When resources are abundant, E. coli can divide every 20 minutes. Prokaryotes generally have just one circular chromosome. The E. coli chromosome is 1.6 millimeter in diameter, making the unfolded circle 100 times greater than the size of the cell. The molecule is packaged by folding in on itself with the aid of basic proteins that associate with the acidic DNA. Circular chromosomes appear to be characteristic of all prokaryotes. The prokaryotes have a site called ori, where DNA replicates, begins, and a site where it ends. Ori is short for origin of replication. Ter is short for terminus of replication. As DNA replicates, each of the two resulting DNA molecules attaches to the plasma membrane. As the bacterium grows, new plasma membrane is added between the attachment points, and the DNA molecules are moved apart. Cytokinesis, which is cell partitioning, begins around 20 minutes after chromosomes duplication is completed. A pinching of the plasma membrane to form a constricting ring separates the one cell into two, each with a complete chromosome. A tubulin-like fiber is involved in the burst ring constriction. Eukaryotic cells divide by mitosis or meiosis. All reproduction involves reproduction signals, DNA replication, segregation, and cytokinesis. Unlike prokaryotes, eukaryotic cells do not constantly divide whenever environmental conditions are adequate, although unicellular eukaryotes do so more often than the cells of multicellular organisms. Some differentiated cells of multicellular organisms rarely or never divide. Signals to divide are related to the needs of the entire organism, not simply the opportunity created by resources. Eukaryotes usually have many chromosomes. Eukaryotes have a nucleus which must replicate and, with few exceptions, divide during cell division. Mitosis generates two cells with the same genetic information as the original cell. Meiosis is a specialized cell division used for sexual reproduction. The genetic information of the chromosomes is shuffled, and the cells, called gametes, typically get one half of the original DNA complement. Going on to the next section is D1, mitosis. Mitosis is the process by which a eukaryotic cell separates the chromosomes in its cell nucleus into two identical sets in two daughter nuclei. It is generally followed immediately by cytokinesis, which divides the nuclei, cytoplasm, organelles, and cell membrane into two daughter cells containing roughly equal shares of these cellular components. Mitosis and cytokinesis together define the mitotic M phase of the cell cycle, the division of the mother cell into two daughter cells, genetically identical to each other and to their parent cell. Mitosis occurs exclusively in eukaryotic cells, but occurs in different ways in different species. For example, animals undergo an open mitosis, whereas the nuclear envelope breaks down before the chromosomes separate, while fungi such as Aspergillus nidulans and Saccharomyces cerevisiae yeast undergo a closed mitosis where chromosomes divide with an intact cell nucleus. Prokaryotic cells which lack a nucleus divide by a process called binary fission. The process of mitosis is complex and highly regulated. The sequence of events is divided into phases corresponding to the completion of one set of activities and the start of the next. These stages are prophase, prometaphase, metaphase, anaphase, and telophase. During the process of mitosis, the pairs of chromosomes condense and attach to fibers that pull the sister chromatids to opposite sides of the cell. The cell then divides in cytokinesis to produce two identical daughter cells. Because cytokinesis usually occurs in conjunction with mitosis is often used interchangeably with mitotic phase. However, there are many cells where mitosis and cytokinesis occurs separately, forming single cells with multiple nuclei. This occurs most notably among the fungi and slime molds, but is found in various different groups. Even in animals, cytokinesis and mitosis may occur independently. For instance, during certain stages of fruit fly embryonic development, errors in mitosis can either kill a cell through apoptosis or cause mutations that may lead to cancer. Next is an overview. The primary result of mitosis is the division of the parent cell's genome into two daughter cells. The genome is composed of a number of chromosomes, complexes of tightly coiled DNA that contain genetic information vital for proper cell function. Because each resultant daughter cell should be genetically identical to the parent cell must make a copy of each chromosome before mitosis. This occurs during S phase. In interphase, the period that precedes the mitotic phase in the cell cycle, where preparation for mitosis occurs. Each new chromosome now contains two identical copies of itself, called sister chromatids, attached together in a specialized region of the chromosome known as the centromere. Each sister chromatid is not considered a chromosome in itself, and a chromosome does not always contain two sister chromatids. In most eukaryotes, the nucleus envelope that separates the DNA from the cytoplasm disassembles. The chromosomes align themselves in a line spanning the cell. Microtubules, essentially miniature strings, splay out from opposite ends of the cell and shorten, pulling apart the sister chromatids of each chromosome. As a matter of convention, each sister chromatid is now considered a chromosome, so they are renamed sister chromosomes. As the cell elongates, corresponding sister chromosomes are pulled toward opposite ends. A nuclear envelope forms around the separated sister chromosomes. As mitosis completes, cytokinesis is well underway. In animal cells, the cell pinches inward where the imaginary line used to be. The pinching of the cell membrane to form the two daughter cells is called cleavage furrow, separating the two developing nuclei. In plant cells, the daughter cells will construct a new dividing cell wall between each other. Eventually, the mother cell will be split in half, giving rise to two daughter cells, each with an equivalent and complete copy of the original genome. Prokaryotic cells undergo a process similar to mitosis called binary fission. However, prokaryotes cannot be properly said to undergo mitosis because they lack a nucleus and only have single chromosomes with no centromeres. Phases of cell cycle and mitosis. The diagram shows mitosis, cytokinesis, cell growth interphase. So when a microtubule connects with the kinetochore, the motor activates using energy from ATP to crawl up the tube towards the originating centrosome. This motor activity, coupled with polymerization and depolymerization of microtubules, provides the pulling force necessary to later separate the chromosomes to chromatids. When the spindle grows to sufficient length, kinetochore microtubules begin searching for kinetochores to attach to a number of kinetochore microtubules find and interact with corresponding non-kinetochore microtubules from the opposite centrosome to form the mitotic spindle period. Prometaphase is sometimes considered part of a prophase. Now going back after the phases of cell cycle mitosis, we have a little bit of discussion to do. Now the next phase is interphase. The mitotic phase is a relatively short period of the cell cycle. It alternates with the much longer interphase where the cell prepares itself for cell division. Interphase is divided into three phases: G1 first gap, S for synthesis, G2 is second gap, and during all these phases the cell grows by producing the proteins and cytoplasmic organelles. However, chromosomes are replicated only during the S phase. Thus, the cell grows G1, continues to grow as it duplicates its chromosomes S, grows more and prepares for mitosis G2 and divides M. Pre-prophase. In plants cells only, prophase is preceded by a pre-prophase stage. In highly vacuolated plant cells, the nucleus has to migrate into the center of the cell before mitosis can begin. This is achieved through the formation of a fragmentosome, a transverse sheet of cytoplasm that bisects the cell along the future plane of cell division. In addition to fragmentosome formation, pre-prophase is characterized by the formation of a ring of microtubules and actin filaments called pre-prophase band underneath the plasma membrane around the equatorial plane of the future mitotic spindle and predicting the position of cell plate fusion during telophase. The cells of higher plants, such as the flowering plants, lack centrioles. Instead, spindle microtubules aggregate on the surface of the nuclear envelope during prophase. The pre-prophase band disappears during nuclear envelope and disassembly and spindle formation in prometaphase. The prophase, normally the genetic material in the nucleus, is in a loosely bundled coil called chromatin. At the onset of prophase, chromatin condenses together into a highly ordered structure called a chromosome. Since the genetic material has already been duplicated earlier in S phase, the replicated chromosomes have two sister chromatids bound together at the centromere by the cohesion complex. Chromosomes are visible at high magnification through a giant microscope. Close to the nucleus are two centrosomes. Each centrosome, which was replicated earlier independent of mitosis, acts as a coordinating center for the cell's microtubules. The two centrosomes nucleate microtubules, which may be thought of as cellular ropes or poles, by a polymerizing soluble tubulin present in this cytoplasm. Molecular motor proteins create repulsive forces that will push these centrosomes to opposite side of the nucleus. The centrosomes are only present in animals. In plants, the microtubules form independently. Some centromeres contain a pair of centrioles that may help organize microtubule assembly, but they are not essential to formation of the mitotic spindle. Prometaphase. The nuclear envelope disassembles and microtubules invade the nuclear space. This is called open mitosis, and it occurs in most multicellular organisms. Fungi and some protists, such as algae or trichomonads, undergo variation called closed mitosis, where the spindle forms inside the nucleus or inside microtubules are able to penetrate an intact nuclear envelope. And each chromosome forms two kinetochores at the centromere, one attached to at each chromatid. A kinetochore is a complex protein structure that is analogous to a ring for the microtubule hook. It is the point where microtubules attach themselves to the chromosomes. Although the kinetochore structure and function are not fully understood, it is known that it contains some form of molecular motor. Now, going to the next page. When a microtubule connects with the kinetochore, the motor activates, using energy from ATP to crawl up the tube toward the originating centrosome. This motor activity, coupled with polymerization and depolymerization of microtubules, provides the pulling force necessary to later separate the chromosomes to chromatids. When the spindle grows to sufficient length, kinetochore microtubules begin searching for kinetochores to attach to. A number of kinetochore microtubules find and interact with corresponding kinetochore microtubules from the opposite centrosome to form the mitotic spindle. Prometaphase is sometimes considered part of prophase. Now this diagram, take a screenshot and a printout and make your inferences. Metaphase, as microtubules find and attach to kinetochores in prometaphase, the centromeres of the chromosomes convene along the metaphase plate or equatorial plane, and the imaginary line that is equidistant from the two centrosome poles. This event alignment is due to the counterbalance of the pulling powers generated by the opposing kinetochores, analogous to a tug-of-war between people of equal strength. In certain types of cells, chromosomes do not line up at the metaphase plate and instead move back and forth between the poles randomly, or they roughly lining up along the midline. Metaphase comes from Greek meta, meaning after. M-E-T-A. Mu is for M, epsilon for E, theta for T, and alpha for A. Right. Because proper chromosome separation requires that every kinetochore be attached to a bundle of microtubules, spindle fibers, it is thought that unattached kinetochores generate a signal to prevent premature progression to anaphase with all chromosomes being aligned. The signal creates a mitotic spindle checkpoint. Anaphase, when every kinetochore is attached to a cluster of microtubules and the chromosomes have lined up along the metaphase plate, the cell proceeds to anaphase, from the Greek eva meaning up, alpha, v alpha meaning up against back or re. Two events then occur. First, the proteins that bind sister chromatids together are cleaved, along with allowing them to separate. These sister chromatids turn sister chromosomes are pulled apart by shortening kinetochore microtubules and move towards the respective centrosomes to which they are attached. And next, the kinetochore microtubules elongate, pushing the centrosome and the set of chromosomes to which they are attached apart to opposite ends of the cell. The force that causes the centrosomes to move towards the ends of the cell is still unknown, although there is a theory that suggests that rapid assembly and breakdown of microtubules may cause this movement. And these two stages are sometimes called early and late anaphase. Early anaphase is usually defined as a separation of the sister chromatids, while late anaphase is the elongation of the microtubules and the microtubules being pulled apart, farther apart. At the end of the anaphase, the cell has succeeded in separating identical copies of the genetic material into two distinct populations. Telophase. Telophase, from the Greek meaning end, is a reversal of prophase and prometaphase events. It cleans up the after effects of mitosis. At telophase, the kinetochore microtubules continue to lengthen, elongating the cell even more. Corresponding sister chromosomes attach at opposite ends of the cell. A new nuclear envelope using fragments of the parent cell's nuclear membrane forms around each set of separated sister chromosomes. Both sets of chromosomes, now surrounded by new nuclei, unfold back into chromatin. Mitosis is complete, but cell division is not yet complete. Next is cytokinesis. Cytokinesis is often mistakenly thought to be the final part of telophase. However, cytokinesis is a separate process that begins at the same time as telophase. Cytokinesis is technically not even a phase of meiosis, but rather a separate process necessary for completing cell division. In animal cells, a cleavage furrow pinch containing contractile ring develops where the metaphase plate used to be, pinching of the separated nuclei. In both animal and plant cells, cell division is also driven by vesicles derived from the Golgi apparatus, which move along microtubules to the middle of the cell. In plants, this structure coalesces into a cell plate at the center of the phragmoplast and develops into a cell wall, separating the two nuclei. The phragmoplast is a microtubule structure, typically for higher planes, whereas some green algae use a phycoplast microtubule array during cytokinesis. Each daughter cell has a complete copy of the genome of its parent cell. The end of cytokinesis marks the end of the M phase. Significance. The importance of mitosis is the maintenance of the chromosomal set. Each cell formed receives chromosomes that are alike in composition and each equal in number to the chromosomes of the parent cell. Transcription is generally believed to cease during mitosis, but epigenetic mechanisms such as bookmarking function during this stage of the cell cycle to ensure that the memory of what genes were active prior to entry into mitosis are transmitted to the daughter cells. Consequence of errors. Although errors in mitosis are rare, the process may go wrong, especially during early cellular division in the zygote. Divisions in the zygote. Mitotic errors can be especially dangerous to the organism because future offspring from this parent cell will carry the same disorder. In non-disjunction, a chromosome may fail to separate during anaphase. One daughter cell will receive both sister chromosomes and the other will receive none. This results in the former cell having 3 chromosomes coding for the same thing, 2 sisters and a homologue, a condition known as trisomy, and the latter cell having only the chromosome, the homologous chromosome, condition known as monosomy. These cells are considered an euploidic cells, and these abnormal cells can cause cancer. Mitosis is a traumatic process. The cell goes through dramatic changes in outer structure. Its organelles disintegrate and reform in a matter of hours, and chromosomes are jostled constantly by probing microtubules. Occasionally, chromosomes may become damaged. An arm of the chromosome may be broken and the fragment lost, causing deletion. The fragment may incorrectly reattach to another non-homologous chromosome, causing translocation. It may reattach to the original chromosome but in reverse orientation, causing inversion, or it may be treated erroneously as a separate chromosome, causing chromosomal duplication. The effect of these genetic abnormalities depends on the specific nature of the error. It may range from no noticeable effect, cancer induction, or organism's death. Endomitosis. Endomitosis is a variant of mitosis without nuclear or cellular division, resulting in cells with many copies of the same chromosomes, occupying a single nucleus. This process may also be referred to as endoduplication, and the cell is endoploid. An example of a cell that goes through endomitosis is the megakaryocyte and yeast. The next section will be D2, meiosis. In biology or life science, meiosis, pronounced meiosis, is a process of a reduction division in which the number of chromosomes per cell is cut in half. In animals, meiosis always results in the formation of gametes. The word meiosis comes from the German verb meion, meaning to make small, since it results in a reduction in chromosome number in the gamete cell. Meiosis is essential for sexual reproduction and therefore occurs in all eukaryotes, including single-cell organisms that reproduce sexually. A few eukaryotes, notably the benthiloid rotifers, have lost the ability to carry out meiosis and have acquired the ability to reproduce by pathogenesis. Meiosis does not occur in archaea or bacteria, which reproduce via asexual processes such as mitosis or binary fission. Each cell has half the number of chromosomes as the parent cell. During meiosis, the genome of a diploid germ cell, which is composed of long segments of DNA packaged into chromosomes, undergoes DNA replication followed by 2 rounds of division, resulting in 4 haploid cells. Each of these cells contain 1 complete set of chromosomes, or half of the genetic content of the original. If meiosis produces gametes, these cells must fuse during fertilization to create a new diploid cell or zygote before any new growth can occur. Thus, the division mechanism of meiosis is a reciprocal process to the joining of 2 genomes that occurs at fertilization. Because the chromosomes of each parent undergo genetic recombination during meiosis, each gamete, and thus each zygote, will have a unique genetic blueprint encoded in its DNA. Together, meiosis and fertilization constitute sexuality in the eukaryotes and generate genetically distinct individuals in populations. In all plants and in protists, meiosis results in the formation of haploid cells that can divide vegetatively without undergoing fertilization. In these groups, gametes are produced by mitosis. Meiosis uses many of the same biochemical mechanisms employed during mitosis to accomplish the redistribution of chromosomes. There are several features unique to meiosis, most importantly the pairing and genetic recombination between homologous chromosomes. History. Meiosis was discovered and described for the first time in CSIR-NET in 1876 by noted German biologist Oskar Hertwig in 1849 to 1922. It was described again in 1883 at the level of chromosomes by Belgian zoologist Edward van Beneden, 1846 to 1910, in Ascaris worms eggs. The significance of meiosis for reproduction and the inheritance, however, was described only in 1890 by German biologist August Weismann, 1834 to 1914, who noted that 2 cell divisions were necessary to transform 1 diploid cell into 4 haploid cells if the number of chromosomes had to be maintained. In 1911, the American geneticist Thomas Hunt, Morgan, 1866 to 1945, observed crossover in Drosophila melanogaster, meiosis, and provided the first true genetics. Evolution. Meiosis is thought to have appeared 1.4 billion years ago. The only supergroup of eukaryotes which does not have meiosis in all organisms is excavata. The other 5 major supergroups, Opisthokonts, Amoebozoa, Rhizaria, Archaeplastida, and chromalveolates, all seem to have genes for meiosis universally present, even if not always functional. Some excavata species do have meiosis, which is consistent with the hypothesis that excavata is an ancient paraphyletic grade. An example of eukaryotic organism in which meiosis does not exist is euglenoid. And the next will be occurrence of meiosis in eukaryotic lifestyles. Meiosis occur in eukaryotic lifestyles involving sexual reproduction, comprising of the constant cyclical process of meiosis and fertilization. In multicellular organisms, there is an intermediary step between the diploid and haploid transition where the organism grows. The organism will then produce the germ cells that continue in the life cycle. The rest of the cells, called somatic cells, function within the organism and will die with it. Cyclic meiosis and fertilization events produces a series of transitions back and forth between alternating haploid and diploid states. The organism phase of the life cycle can occur either during the diploid state gametic life cycle or during the haploid state zygotic life cycle, or both. A sporic life cycle in which there are 2 distinct organisms facing one during the haploid state and the other during the diploid state. In this sense, there are 3 types of life cycles that utilize sexual reproduction, differentiated by the location of the organism's phases. In the gametic lifestyle, the species is diploid, grown from a diploid cell called the zygote. In the zygotic lifestyle, the species is haploid instead, spawned by the proliferation and differentiation of a single haploid cell called the gamete. Humans, for example, are diploid creatures. Human stem cells undergo meiosis to create haploid gametes, which were spermatozoa for males or ova for females. These gametes then fertilize in the fallopian tubes of the female, producing a diploid zygote. The zygote undergoes progressive stages of mitosis and differentiation, turns into a blastocyst, and then gets implanted in the uterus endometrium to create an embryo. In the gametic life cycle of which humans are a part, the living organisms is diploid in nature. Here we will generalize the example of human reproduction and state previously. The organism's diploid germline stem cells undergo meiosis to create haploid gametes, which fertilize to form the zygote. The diploid zygote undergoes repeated cellular division by mitosis to grow into the organism. Mitosis is a related process to meiosis that creates 2 cells that are genetically identical to the parent cell. The general principle is that mitosis creates somatic cells and meiosis creates germ cells. Come again? The general principle is that mitosis creates somatic cells and meiosis creates germ cells. In the zygotic life cycle, the living organism is haploid. 2 organisms of opposing gender contribute to haploid germ cells to form a diploid zygote. The zygote undergoes meiosis immediately, creating 4 haploid cells. These cells undergo mitosis to create the organism. Many fungi and many protozoa are members of the zygotic life cycle. Finally, in the sporic life cycle, the living organism alternates between haploid and diploid states. Consequently, this cycle is also known as the alternation of generations. The diploid organism's germline cells undergo meiosis to produce gametes. The gametes proliferate by mitosis, growing into haploid organisms. The haploid organism's germ cells then combine with another haploid organism's cells, creating the zygote. The zygote undergoes repeated mitosis and differentiates to become the diploid organism again. The sporic life cycle can be considered a fusion of the gametic and zygotic life cycle. Interphase is divided into 3 phases. Growth 1, that is G1 phase, and immediately follows cytokinesis. This is a very active period where the cell synthesis is synthesizes its vast array of proteins, including their enzymes and structural proteins it would need for growth. In G1 stage, each of the 46 human chromosomes consists of a single, very long molecule of DNA. At this point, cells are 46 2n, identical to somatic cells. Synthesis S phase, the genetic material is replicated. Each of its chromosomes duplicates 46 2n. The cell is still haploid. However, because it still contains the same number of centromeres. However, the identical sister chromatids are in the chromatin form because spiralization and condensation into denser chromosomes have not taken place yet. It would take place in prophase 1 meiosis. Growth 2 or G2 phase, which is absent in meiosis. Interphase is immediately followed by meiosis 1 and meiosis 2. Meiosis 1 consists of segregating the homologous chromosomes from each other, then dividing the diploid cell into 2 haploid cells, each containing one of the segregates. Meiosis 2 consists of decoupling each chromosome's sister strands, chromatids, segregating the DNA into 2 sets of strands, each set containing one of each homolog, and dividing both haploid, duplicated cells to produce 4 haploid, unduplicated cells. Meiosis 1 and 2 are both divided into prophase, metaphase, anaphase, and telophase subphases, similar in purpose to their analogous subphases in the mitotic cell cycle. Therefore, meiosis encompasses the interphase G1, S, and G2, meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. And meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. Meiosis generates genetic diversity in 2 ways. 1, independent assortment of chromosomes at both of the meiotic divisions allows genetic differences among gametes. And 2, physical exchange of chromosomal regions by homolog recombination during prophase 1 results in new genetic combination within chromosomes. Meiosis 1. In meiosis 1, the homologous pairs in a diploid cell separate, producing haploid cells 46n. The 46 chromosomes number is significant, and a regular diploid cell contains 46 chromosomes and is considered 2n because it contains 23 pairs of homologous chromosomes. However, after meiosis 1, although the cell contains 46 chromosomes, it is only considered N because later in anaphase 1, the identical sister chromatids will remain together as the spindle pulls the pair towards the pole of the new cell. In meiosis 2, a process similar to mitosis will occur, whereby the sister chromatids are finally split, creating 2 haploid cells, 23N. Prophase 1, homologous chromosomes pair and crossing over or recombination occurs as a step unique to meiosis. Chromosome form structures and are called synapses. The paired chromosomes are called bivalents or tetrads, which have 2 chromosomes and 4 chromatids, with 1 chromosome common from each parent. At this stage, non-sister chromatids may cross over at points called chiasmata. Leptotene. The first stage of prophase 1 is the leptotene stage, known as leptonema, from Greek words meaning thin threads. During this stage, individual chromosomes begin to condense into long strands within the nucleus. However, the 2 sister chromatids are still so tightly bound that they are indistinguishable from one another. The chromosomes in the leptotene stage show a specific arrangement where the telomeres are oriented towards the nuclear membrane, hence the stage is called bouquet stage. As zygotene. The zygotene stage, also known as zygonema, from Greek words meaning paired threads, occurs as the chromosomes approximately line up with each other into homologous chromosomes. The combined homologous chromosomes